Hello everyone, welcome to the new session of Biochemistry of Proteins. So in this session we are going to study about proteins. So in this session we are having one introduction to protein. So we will study about protein, what is the protein, what is the structure, what it is made up of, right? So all this we are going to cover up in the introduction of proteins. Then there will be a general characteristic of protein. So we are having some characteristic, right? And that we are going to study about the proteins. Then different folding of protein, we will have primary, secondary, tertiary and quaternary structure of protein, so that we will see here. Okay, then after that we will have some properties of protein. So according to that, different proteins are has been classified, so that also we are going to cover up in this session. So let us start. The term protein was coined by Berzelius in 1838. So Berzelius coined first time proteins, right? This is the term coined by Berzelius in 1838, and a significant role in the structural and the metabolism of the cell protein. So this Berzelius also said that this particular protein is having two very important things. One is the structural component, they form the structural component of the cell, right? So can we guess anything that it is made up of proteins in a cell? Structural component of protein, right? So any guess? So I'll tell you about the plasma membrane, right? The plasma membrane is the outer covering of the animal cell, whereas in the case of plant cell, beside cell membrane there is also the cell wall, right? But this plasma membrane, if we talk about the plasma membrane, then it is made up of lipid bilayer. So here we can see the lipid bilayer in this manner, right? Lipid bilayer, and in these lipid bilayer there are some transmembrane like this protein structure. This is the protein. So around 50 to 52% of this any of the cell plasma membrane is made up of protein. Rest of it is the uh, lipid, right? So 12% of the cell content, if we talk about the total cell content of any cell, then it is made up of protein, and 75% dry weight. And if any we can dry any of the component of the cell, then you will get 75% of its dry weight. Proteins are highly specific in nature. They have the specific shape and structure. So if we talk about the shape, structure and surface, then they are very specific. So the protein which is formed in a form of polypeptide, it get folding like this, and this folding is not a random, it is very specific. And if this folding is specific, then only they will have the specific structure of any protein is very specific, its shape is very specific, and its surface is very specific. All these things are dependent upon the folding of proteins. So we will see in the coming slide that how the proteins are get folded, right? So the unique structure that enables it to carry out a specific function, and all these specific structure, specific shape and specific surface of any protein provide them a specific function. So they perform a specific function because they contain a specific structure, specific shape and surface as well, right? And how this they achieve this, all these three components like structure, shape and surface, by the folding, specific folding of proteins. General characteristic of proteins. So let us talk about what are the general characteristics of proteins. Chemically, a protein is made up of carbon, hydrogen, nitrogen, oxygen and sulfur. So if you see the structure of protein, okay, and chemical analysis of any of the protein, then you will see that this is made up of carbon, hydrogen, nitrogen, oxygen and sulfur. So not all the proteins are containing the sulfur, but yes, there are some sulfur containing amino acids, and if that amino acid is present in uh, protein, then that protein will have the sulfur as well. Some proteins additionally contain phosphorus, iron and other elements. So there are some other proteins also, they contain phosphorus, iron, right? Proteins are variously folded linear heteropolymers of the amino acids. So what are the building blocks of any protein? So building blocks of any proteins is amino acid. So how this amino acids comes in the cell? We eat proteins, we eat animal protein, we eat plant protein, and that protein get digested in our uh, digestive tract. That is, this is start from the stomach and it ends in the small intestine, right? And all these digested protein get absorbed in the form of amino acids. Now these amino acids are present in the blood. It get assimilated, right, and reaches to every cell. And when it reaches to every cell, cell utilize these amino acids to make it specific proteins, right? So this is very, very important. And every protein is having a definite shape, structure and surface, and that they achieved by a specific folding pattern. The linear polymers of amino acids are called as polypeptides. So here what we see that there is a mono that they are having the monomer, right, like this, and each monomer is what? Amino acid. And when amino acids are attached in this in a form of linear fashion like this, having these are the peptide bonds. So this is called polypeptide. Polypeptides, right? So we will see more about it and then we are going to understand that the amino acids are linked serially by the peptide bonds CONH found between the amino group of one amino acid and carboxylic group of adjacent. So here what we see that suppose one of the amino acid is providing this NH2 group and another amino acid is providing is COOH group like this, right? So this is a chiral carbon through which the NH2 is attached, and here this amino acid, this is amino acid 1, this is amino acid 2, right? So what will happen? Water is eliminated out here, H2O, this is eliminated out, and what they form? NHOC like this. Okay, so in this way they form peptide bond. Okay, they form the peptide bond. The sequence of amino acid present in a polypeptide is specific for the particular protein. Now this amino acid 1, amino acid 2, it is pre-decided for any particular type of protein. Right? So it is not a random that any protein, any amino acid can comes and they get a peptide bond and they will form. Okay, and so if we are talking about protein 1, then protein 1 will have a sequence, a specific sequence of amino acid like this. and these amino acids are fixed, and if any another amino acid by mistake comes, then this particular protein may not get a proper folding, and its structure, shape and surface may get altered, and in that case this protein will not perform a particular function. So now you can understand how specific it is. Every protein is having its own specific structure, and that depends upon the amino acid which are present in this sequence. Okay, the distinctive sequence of amino acid unit is governed by the codon sequence, the gene of cistron that control its formation. So where this codon is present, so basically the codon is present on the DNA. From there, when transcription takes place, so on the template of DNA, mRNA gets synthesized. Okay, from DNA mRNA gets synthesized. So this is the mRNA. Now on this mRNA, what will happen? Protein gets synthesized with the help of ribosomes like this. These ribosomes will help to form the proteins. Okay. So this protein is studying what? It is studying mRNA. This ribosome is studying mRNA. What is the sequence? This is present on the mRNA. And from where this mRNA got its sequence? It got its sequence from DNA. So there comes the codon sequences, the gene of cistron that control the formation, right? Proteins are incredibly diverse in their functions and are involved in nearly every aspect of cellular and organismal body. So, proteins are incredibly diverse. Incredibly diverse means every protein is having its own particular structure. So that is why we are saying that they are incredibly diverse, and every protein perform a particular functions, right? So in the respect of function also, they are very diverse, and they are involved in nearly every aspect of cellular and organismal body. Means cellular structure, they are forming a cellular structure, as well as many cellular functions are involved these proteins, because we all know that proteins are basically enzymes. Also enzymes, chemical composition nature is protein, and we see that in the biology, all the reactions which are taking inside the living body is influenced by a particular enzymes, right? So this is very, very important. Means every reaction is taking place with the help of enzyme, and that is protein. 20 amino acids are used, right, in the synthesis of all types of proteins. So we see that there are so many types of protein, and all the proteins are made up of just 20 amino acids, right? Collagen is the most abundant protein in the animal world. So if we talk about the animal world, then we see that collagen is the most abundant protein in the animal world, whereas the rubisco, that is the ribulose bisphosphate carboxygenase, right, it is not only the most abundant protein in the plant but also whole biosphere. So now we talk about the levels of protein structure. So we will see how the proteins are forming its a definite structure. So there are four different conformation, right? Four levels of protein folding, four different conformation we can say: primary structure, secondary structure, tertiary structure, and quaternary structure. And then the functional protein will form, right? So to form a particular function, every protein has to pass these levels. So first of all, they will form the primary structure of protein, then they will form the secondary structure of protein, then they will form the tertiary structure of protein, and then they will form the quaternary structure of protein. So primary structure, it is the description of basic structure of protein, right? So what we see, we see that basic structure of protein, primary structure, how it is formed. Suppose this is the amino acid 1, then this is the amino acid 2, then this is the amino acid 3, this is amino acid 4. I'm just taking the 4 example here, and what we see, they are having the bond in between them. And what type of bond are these? These are the peptide bond. Peptide bond, right? And so this is showing what? This is the primary structure of protein, right? Primary structure of protein. So this include the number of polypeptides, number and sequences of amino acid in each polypeptide. So you know what is the sequence of amino acid, what is amino acid number 1, what is amino acid number 2, what is amino acid 3, what is 4. You all know this. And that gives you the sequence of polypeptide, right? And when you know the number of amino acid in a polypeptide, when you know the sequence of amino acids polypeptide, right, then you get the primary structure of protein. Specific amino acid determine the places where the polypeptides are to be bent or fold and where the different length will be attracted to each other, right? So specific amino acid determines the places where these polypeptides are to be bent and where to get fold and where the different length will be attracted to each other. So this is actually the primary structure of the protein. So here we can see the distance between the two adjacent peptide bond is about 0.35 nm. So if we see what is the distance between these two, you can say this is the COH group of one amino acid, this is NH2 of another amino acid, and there is a peptide bond in between, right? And what is the length of this peptide bond? This is 0.35 nm. Okay, and the first amino acid is this is the N, while the last amino acid that ends represented by carboxyl group, and that is denoted by C. Secondary structure. It is the development of the new steric relationship of amino acid present in the linear sequence inside the polypeptide, right? What does the secondary structure show? The secondary structure is formed by the new steric relationship of amino acid present. Now, if we are having a polypeptide like this, right? Now, these polypeptide will have some interaction, these amino acid will have some interaction in between, right? And that helps to form a particular structure, right? So, some of the new relationships of the regular nature and give periodicity to the structure. So, in this way, they form a different form of structure. Earlier, we are having in the primary structure just a single polypeptide. Now, these polypeptide will form a helix-like structure because in the polypeptide, in this linear polypeptide, when it become very long, it get folded in the form of helix. Now, this helix is not in a random folding. The amino acid which is present here will have interaction with this, and it will form a helical structure like this. So you can say that these helical structures are very specific and some of the new relationships of a regular nature and give periodicity to the structure. There are three types of secondary structures. Helix, that is the a helix, that is the alpha helix or A helix, beta pleated sheet, this is the beta pleated sheet, and third one is the collagen helix. So three different types of secondary structure we are having. Let us see what are those. The alpha helix is a polypeptide chain is a coiled spirally, generally in the right-handed manner. So this alpha helix is a polypeptide chain, right? It is a polypeptide chain and it is coiled spirally like this. They coiled spirally and form a, and this coiling is in which left or right? It is right-handed manner. It is in the right-handed manner. At places, the helix is less regular, forming a random coils. But at some point, you can see this coil is very random. But at some point, this is again forming a alpha helix. And this alpha helix is stabilized by the hydrogen bond between the oxygen of the carboxyl group of one amino acids residue and NH group of the next fourth amino acid. So suppose this is the first amino acid here and this is the fourth amino acid here. So they will have hydrogen bonding in between. And this helix structure is get stabilized in this way due to the hydrogen bonding. So secondary structures form in this way. Now here we can see the alpha helix is often involved in the protein-protein interaction. Now where we can see this protein-protein interaction? Any guess? So the protein, one of the protein is like this. Now another protein get attached to it. Now this attachment is because of this protein-protein interactions, because here it is also having the alpha structure and here it is also having the alpha structure. DNA binding. So DNA is there. Any protein goes and bind to it. Because you know, in the process of replication, DNA polymerase, okay, gyrase, SSB protein, all these are the proteins, right? And they goes to bind the DNA. So that is the DNA binding protein. So what this protein is having, what type of secondary structure? It is having alpha helix and other structural functions. Now the beta sheet can be found, another form of secondary structure. This is the first form of secondary structure. Alpha helix is the first form of secondary structure. Now the beta sheet, beta pleated sheet, in fact, the secondary structure, second form of secondary structure, means you can say the another type of secondary structure. So the beta sheets can be found in a various protein structure and can be involved in the forming the core of proteins or providing the structural stability. So here you can see this sheet like structure. One sheet is like this, another sheet is like this, another is this, like this. So they are having this beta pleated sheet like structure, and this is the alpha helix. This is the alpha helix and this is the beta pleated sheet, and both are considered as secondary structure of proteins. The beta pleated sheet structure, two or more polypeptide chain get interconnected by the hydrogen bonding. So in the beta pleated sheet, this is the structure. Now this is again this is like this. This is like this. So they are forming the sheet like structure, and this sheet like structure get stabilized by the hydrogen bonding. So what type of interaction taking place in the formation of the secondary structure? Either it is a alpha helix, either it is a beta pleated sheet. Hydrogen bonding is very, very important for its formation. Adjacent strands of polypeptides may run in the same direction, that is parallel beta sheet, that is the beta keratin, or opposite directions, anti-parallel beta sheet. Suppose this is one of the strand and this is another strand, right? Both are in one direction and they are having the hydrogen bond in between. Then this is called as a parallel beta sheet. Okay, means the two polypeptides are running in the same direction. And this type of, we see the structure in the beta carotene. And sometimes we see in the opposite direction, like this, opposite direction. And that is called as anti-parallel beta sheet. Anti-parallel. This is the anti-parallel. And this is the parallel beta sheet. And this we can see in the fibroin of silk. This parallel beta sheet we see in the case of beta carotene, whereas in the this occur in the opposite direction, that is the anti-parallel. We can see it is in the fibroin of silk. Now, the beta pleated proteins are more extended than the ones having the alpha helix structure. Okay, so the beta pleated proteins are more extended than the ones having the alpha helix structure. So this is about the secondary structure. Now Ramachandran, this is the very important part. The Ramachandran discovered that there are generally 3 strands of polypeptides coiled around the other. One coiling, another coiling, and there is one more coiling, like this. Okay, so he discovered that 3 strands of polypeptide coiled around one another, and that coil is strengthened by the establishment of hydrogen bond between NH group of glycine residue, each strand with COH group of the other strand, and that we form the collagen, right? So let us see what is the difference between the alpha helix structure and beta pleated sheet. So this, if we talk about the alpha helix structure, then can exist as a right-handed or a left-handed helical structure, whereas the beta pleated sheet can exist as a parallel or anti-parallel sheet. So the two form here. Now, the hydrogen bond formed between the carbonyl of the first amino acid would form a hydrogen bond with NH of the fifth amino acid. So, if they are forming the alpha helix like this, so suppose here it is amino acid 1, then the fifth amino acid which is present here, they will have the hydrogen bond in between and arrange in the parallel and anti-parallel. We have already discussed that is in the case of parallel, and here we can in the case of anti-parallel. Polypeptide when are running and they are having hydrogen bond in between. It forms the continuous stretch of polypeptide, whereas it does not form the continuous stretch of polypeptide. Examples of alpha helix structure, that is the keratin, fibrin, myosin and the tropomyosin. So myosin and tropomyosin, if you remember, it is present in the muscle contraction we have studied. And fibroin, that is the silk thread secreted by the spiders and insects, is an example that is having the beta pleated sheet. Now comes the tertiary structure. The overall 3-dimensional structure of a polypeptide is called its tertiary structure. The tertiary structure is primarily due to the interactions between the R group of the amino acid. Now we have seen the secondary structure. After that, we can see the tertiary structure formations, right? So there is the further folding of protein and they will form the tertiary structure. This structure is more common in the globular proteins. So for the formation of tertiary structure, these are the bonds that we find: ionic bond formations, hydrogen bond formation, disulfide bond formation, hydrophobic bond formation, right? And example, myoglobin and many proteinaceous enzymes. So myoglobin is one of the examples that you can see for the tertiary structure, right? So in the formation of tertiary structures, so many interactions are involved. These are the types of bonds you can see at the tertiary level of protein structure. Okay, after this, now comes the quaternary structure. This structure exists in the protein with more than one polypeptide chain. If one polypeptide chain is forming a particular like this, then there will be one more polypeptide. Now in these two polypeptide there will be some interactions. Okay, so this is showing the tertiary level structure and this is again showing the tertiary level structure. Now these two polypeptide at a tertiary level structure combine together, means they have some interactions together and finally they form the quaternary structure. It means that two different polypeptide at the level of tertiary structure interact with both these polypeptide interact together and form a protein structure of the quaternary level. So each polypeptide chain function as a subunit of the protein. Now this is the total protein, this is the total protein, right? And these two are subunits. So subunit is having what level of structure? Tertiary structure. And whole protein, whole functional protein is having what level of structure? Quaternary structure. So the different subunits of a chain are packed together to give a conformation. Hemoglobin, if we see, it is having two alpha chains and two beta chain, right? Two alpha chain and two beta chain, and both these, all these four chains means interact together to form a stable quaternary structure of the hemoglobin. So the different types of bonds are involved in the folding of polypeptides. They are having the covalent bond, they are having the ionic bond, they are having the hydrogen bond, van der Waals interaction, and also the hydrophobic bond. So here you can see the quaternary structure. They are having the ionic bond, right? They are having the disulfide linkage, right? They are having the hydrophobic interaction, and they are also having the hydrogen bond. Now, the properties of proteins, variety. There are thousands of proteins in the different organism and they are having this specificity, and each protein has the certain specific proteins not found in other, right? So, a specific protein is found in a specific organ, and there are some specific proteins that are found in the particular organism only, right? And closely related species share several common proteins. So there are some large sized molecules. The proteins are large sized molecules with a minimum molecular weight, that is 450 to maximum molecular weight of 4,600,000. Properties of protein: colloids. Being large size, they form the colloidal solutions. So the proteins are quite a big size, so they form the colloidal solution. Reactivity: due to the folding of chain, proteins have a specific configuration with the specific reactive group, right? And the permeation, that is the cell membrane, do not allow the permeation to the proteins. Then they are having the denaturation also. So the bonds in the protein is easily broken by the high temperature with high energy radiations like soap, disinfectant, detergents and alcohol. So these are some of the properties of proteins. With that, we completed this chapter. Thank you very much.